So today we'll talk about phototransduction in the visual system. So within the visual system, we have the eye. In the back of the eye, there's the retina. Within the retina, there are three different types of cells: the ganglion cell, the bipolar cell, and the photoreceptor cell. The photoreceptor cell is the most important cell here, as it is where phototransduction takes place. So there are two different types of photoreceptor cells. There are cones and there are rods. They basically do the same thing, so today we'll just focus on one type and we will focus in on the rods. So there are three different parts of the rods: the outer segment, the inner segment, and the synaptic ending. The most important part here would be the outer segment, as is where phototransduction actually takes place. So we'll zoom into the outer segment, and we will first focus on the outer segment when light is introduced to this eye. So here, there will be a light stimulus, which will then hit rhodopsin and activate it. Activated rhodopsin will then stimulate the transducin G protein. In turn, the transducin G protein will activate cGMP phosphodiesterase. This is an enzyme that will break down cGMP. cGMP being broken down will result in reduced level of cGMP. This will result in the closing of cGMP channels, which will inhibit Na from entering the cell. The potassium channels will not be altered and will free flow out of the cell. This in turn will result in a hyperpolarization of the cell. So the inside of the photoreceptor cell will become more negative. This will result in a decreased neurotransmitter release. So going back to the big picture, we will put together what this all means with a decrease of neurotransmitter release. So back here, you can see the three different types of cells. So there will be no neurotransmitter release from the photoreceptor cell. So no neurotransmitter being released will result in no IPSP, which also means inhibitory postsynaptic potential. In turn, the cell will depolarize and neurotransmitters will be released from the bipolar cell onto the ganglion cell. So this will result in an EPSP, an excitatory postsynaptic potential, on the ganglion cell. So in turn, this whole cascade of events will result in an active potential that will travel to the brain to be processed. So now we'll switch over to the dark stage in the photoreceptor cell, where there is no light stimulus. So in turn, rhodopsin will be phosphorylated by a receptor kinase. This will then result in the G protein transducin not being stimulated, will not activate the cGMP phosphodiesterase. Phosphodiesterase not being activated will not break down the cGMP, so there will be an increased level of cGMP. cGMP channels will then not close, and Na will free flow into the cell. The K channels will also not be inhibited, and potassium will be able to flow out of the cell. In addition, there's a sodium-potassium pump operating via active transport to keep the relative balance of ions stable across the cell membrane. And a baseline neurotransmitter release. So now we'll go back to the big picture. So the pathway in the dark, the photoreceptor cell will release neurotransmitters, and there will be an IPSP generated on the bipolar cell. So the membrane potential will be around negative 40 millivolts. This will result in no neurotransmitters being released from the bipolar cell. No neurotransmitters being released onto a ganglion cell from the bipolar cell will result in no action potential and therefore no stimulus being sent to the brain.